Skeletal muscle fibers contract as a result of electrical impulses from nerve cells called motor neurons. To understand how this happens, we divide the entire process into 3 phases. First, the neuron stimulates the muscle fiber. This is covered in the IP 2.0 topic, Muscle Excitation at the Neuromuscular Junction. Second, the muscle fiber action potential leads to muscle fiber contraction. This process is explained in the IP 2.0 topic, Excitation-Contraction Coupling. Third, muscle contraction occurs through cross-bridge cycling, which is the focus of this topic. Remember that excitation-contraction coupling triggers calcium ions binding to troponin, causing the myosin binding sites on actin to become uncovered. Cross-bridge cycling is the sequence of events where the myosin of the thick filaments pulls on actin, which slides the thin filaments toward the center of the sarcomere and results in the shortening of the sarcomere. Multiplied over all the sarcomeres in the myofibrils, this results in muscle contraction. To start, let's magnify a myofibril and examine the features of the sarcomere. Each myofibril is made up of thousands of sarcomeres. The sarcomere is the basic unit of muscle contraction. By understanding how one sarcomere shortens, we can understand how the simultaneous contraction of thousands of sarcomeres along hundreds of myofibrils leads to contraction of one muscle fiber. Each sarcomere consists of an arrangement of thick and thin filaments. The thick filaments are made up of many myosin proteins. Each myosin molecule is shaped a bit like a golf club, with 2 heads and a tail. Each myosin head contains 2 important binding sites. One site binds to ATP, which will supply the energy used for muscle contraction. The second site binds to the myosin binding site on actin. The result is a cross-bridge between thick and thin filaments. Now that we've examined the thick filaments, let's review the structure of the thin filaments. Recall from the IP 2.0 topic, Excitation-Contraction Coupling, that the thin filaments are composed of 3 types of molecules: actin, tropomyosin, and troponin. The actin molecules form a pair of strands that wind around each other. Each actin molecule has a myosin binding site to which a myosin head can attach. When the muscle is not being stimulated to contract, these binding sites are covered by tropomyosin. However, the myosin binding sites are exposed when calcium ions bind to troponin during excitation-contraction coupling. Now that we have identified the main components of the thick and thin filaments, let's examine how they are arranged within the sarcomere. When viewed with a microscope, the skeletal muscle tissue has a striped or striated appearance with alternating light and dark bands. Let's examine what causes these stripes. The lightest areas are called I-bands. The only filaments in the I-band are thin filaments. A Z-disc in the center of each I-band anchors the thin filaments. The Z-discs also mark the ends of the sarcomere. The darker central areas of the sarcomere are the A-bands. The A-band is the region occupied by thick filaments. The thick filaments are anchored to the M-line, which marks the center of the sarcomere. The central region of the A-band is slightly lighter than the rest of the A-band because it contains thick filaments but no thin filaments. This region is called the H-zone. Now that you know the regions of the sarcomere, let's examine the sliding filament mechanism of muscle contraction. Recall that the thin filaments extend from the Z-disc toward the center of the sarcomere, overlapping partially with the thick filaments. According to the sliding filament mechanism, a muscle contracts because the thin filaments slide past the thick filaments toward the center of the sarcomere from both sides. In doing so, they pull the Z-disc at either end of the sarcomere toward the central M-line, shortening the sarcomere. This occurs at all the sarcomeres along the length of the muscle fiber. Let's look at how the bands change during muscle contraction. Notice that the I-band shortens as the sarcomere goes from a relaxed to a contracted state. However, the width of the A-band remains the same length. This is because the filaments themselves do not change in length, they just overlap more. Now let's focus on the H-zone. Like the I-band, the H-zone narrows and may even disappear as the thick and thin filaments overlap more and more. How does this sliding movement occur? During muscle contraction, when myosin heads bind to actin, they form cross bridges. Once cross bridges form, myosin pulls the thin filaments toward the center of the sarcomere. The pulling motion of the myosin heads is similar to how the oars of a rowboat work. The thick filament is the boat, and the oars are the myosin heads that form cross bridges. Each stroke produces only a little movement. However, just as repeated oar strokes move a boat, so the repeated breaking and reforming of myosin cross bridges pulls actin toward the M-line. A single action potential is sufficient to stimulate many cross-bridge cycles. This shortening takes place in all the sarcomeres along the length of each myofibril and in all the myofibrils of a muscle fiber. The result is muscle contraction. Now that we know that thin filaments slide over thick filaments during muscle contraction, let's examine how the process works in detail. Myosin molecules have two important features that enable the thick filament to bind to and move the thin filament. First, the hinge portion of the tail allows the myosin head to reach the binding sites on actin. Second, the myosin head can pivot back and forth between high-energy and low-energy positions. This pivoting motion constitutes the power stroke that pulls the thin filament along the thick filament. A sarcomere shortens when myosin heads on the thick filaments form cross bridges and pull on the actin molecules in the thin filaments. The process known as the cross-bridge cycle consists of 4 steps. 1. Cross-bridge formation. 2. The power stroke by the myosin head. 3. Cross-bridge detachment. And 4. Reactivation of the myosin head. Let's examine each step. Step 1. Cross-bridge formation. The cycle starts when excitation-contraction coupling causes the myosin binding sites on actin to be exposed. Myosin heads bind to these sites, forming cross bridges. Myosin starts in its high-energy state, in which the ATP binding site on the myosin head is occupied by ADP and inorganic phosphate. The myosin head is ready to pivot for the power stroke. Step 2 is the power stroke. Release of inorganic phosphate from the myosin head causes the head to pivot, pulling the thin filament toward the center of the sarcomere. ADP is then released as well. The myosin head is now in its low-energy state, but it remains attached to actin. The energy for the power stroke is ultimately provided by the ATP that binds to the myosin head. Step 3 is cross-bridge detachment. Myosin remains attached to actin until an ATP molecule binds to the empty ATP binding site on the myosin head. This weakens the link between the myosin head and actin, and the myosin head detaches. The detached myosin head remains in its low-energy state. Step 4 is the reactivation of the myosin head. This occurs when the ATP bound to the myosin head undergoes hydrolysis to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, pivoting it back to its high-energy state. The cross-bridge cycle starts again when the energized myosin head forms another cross-bridge. As long as the myosin binding sites on actin remain exposed and ATP is available, the cross-bridge cycle repeats, pulling the thin filaments toward the center of the sarcomere. During muscle contraction, cross-bridge cycles occur between the thick and thin filaments along the entire length of each sarcomere. But not all myosin heads are bound or disconnected at the same time. They can be in different steps of the cross-bridge cycle. Remember that there are thousands of sarcomeres in a myofibril and thousands of myofibrils in each muscle fiber. Cross-bridge cycling at all of these sarcomeres causes the entire muscle to contract. Cross-bridge cycling ends when the concentration of calcium ions in the sarcoplasm decreases to the resting level of the unstimulated muscle. Two events are responsible for this decrease in calcium concentration. First, the terminal cisterns cease releasing calcium ions. This occurs when signals from the motor neurons stop and no additional action potentials conduct along the sarcolemma and T-tubules. Second, calcium ions are actively transported out of the sarcoplasm back into the sarcoplasmic reticulum by specialized calcium pumps. This calcium transport reduces the calcium ion concentration in the sarcoplasm below the threshold necessary for excitation-contraction coupling. The reduced level of calcium ions causes troponin to return to its original shape, causing tropomyosin to glide over and cover the myosin binding sites on actin. Even though some of the myosin heads are in the high-energy state, they can no longer form cross-bridges. The sarcomeres return to their original resting length and the muscle relaxes. Let's review what you've learned. During a muscle contraction, sarcomeres shorten because the thin filaments are pulled past the thick filaments toward the center of the sarcomere. This is called the sliding filament mechanism. The thin filaments are pulled by the myosin heads of the thick filaments, which continuously form and break thousands of cross-bridges with the actin molecules of the thin filaments. Cross-bridge cycling is the sequence of events where myosin pulls on actin. The 4 steps of the cross-bridge cycle are the formation of cross-bridges, the power stroke, cross-bridge detachment, and myosin head reactivation. Cross-bridge cycling continues as long as the myosin binding sites on actin are exposed and ATP is available. Muscle relaxation occurs when the calcium ion concentration drops below its threshold value for binding to troponin. This causes tropomyosin to shift back to cover the myosin binding sites on actin. Cross-bridges are now prevented from forming, so the thin filaments slide back to their resting position. The sarcomere is now ready for the next stimulation at the neuromuscular junction to trigger another muscle contraction.